We'll begin the class with a discussion of how we know that viruses really are distinct infectious agents. That is, how do we know that viruses are a different category entirely from other microscopic things that cause diseases, things like bacteria or a cellular parasite like malaria? The first clue that there was something other than or something smaller than bacteria that causes disease came at the end of the 19th century. At that point, filters had been developed that had pore sizes that were small enough that bacteria would not pass through. Therefore, the filtrate was considered sterile. Dmitry Ivanowski in 1892 was investigating tobacco mosaic disease. And so here you see a leaf of a tobacco plant, and the mottled appearance is indicative of this particular disease. So he took some leaves and he homogenized them, and he passed them through one of these filters, and he knew that bacteria would not pass through the filter. He collected the filtrate, he put it on some healthy plants, and then eventually, very shortly, these plants developed the disease. So we saw that this was something, it had to be smaller than bacteria because he knew that the filter was good. Ivanowski did a good experiment, but he sort of fell down at the last step because he did not make the inference that this was a new infectious agent. What he thought was that it was some sort of a toxin from a bacteria or maybe a bit of cell waste that was causing the disease. So he couldn't actually come to the right conclusion from his data. 6 years later, Martinus Beijerinck in Holland did make the inference from the similar experiment that whatever the material was that passed through the filter was a unique infectious agent. And in Germany, Friedrich Loeffler and Paul Frosch showed that foot and mouth disease, an animal disease of course, was caused by a similar type of agent. So thus it wasn't just a matter of a unique infectious agent of plants. These, whatever they were, existed in a wide variety of organisms. And so how did they make this conclusion? It's because they realized that it couldn't be a toxin because whatever this agent was, it could replicate, or more precisely, it could be replicated. So if you want to test the hypothesis that viruses are infectious, that is, that they can be replicated, you've got to dilute it way out, and so we do this serially. So you dilute it first 1 to 10, and then you dilute the 1 to 10 by 1 to 10 again, so you have a 1 to 100, then 1 to 1,000, and so on. And you show that no matter how small or how tiny the dilution is, how great the dilution is, you still end up with the same disease. It may take more time, but you get the disease. And as a control, you dilute a toxic molecule. So let's just give an example, is methyl isocyanate. We're going to dilute that in the same way. When you do this experiment with virus, you see that no matter how much you dilute the virus, the cells die. And so these are tissue culture cells, of course, because nowadays you would do this in a tissue culture. And the way the cells are sort of rounded up on this plate shows what we call cytopathic effect. That is, they just look like dead cells. With the toxin, at the high concentrations, the cells still die, but at low concentrations, the cells are still alive. So dead cells, right? They're still alive at the farther end because they're nice and flat and they're attached. So the conclusion is that virus will always kill cells, though increasing the dilution increases the time to CPE, that is cytopathic effect. But toxins can be diluted to a point to which there is no effect on cells. And so thus the difference between virus and a poison. However, the name virus dates back to this original idea because virus is Latin for poison. One thing that makes viruses really different from bacteria is that viruses have to be inside living cells in order to be replicated. I'm sure you're familiar with the procedure of putting some bacteria on an agar plate and you leave it alone for a couple of hours to a couple of days and you get nice colonies all over the place. Viruses can't do that. Viruses cannot replicate on nutrients alone. There must be living cells. So either living cells in the form of an experimental host or in the form of tissue culture, that is cells growing either in a dish or growing in suspension. Having said that, not all viruses can be amplified in experimental systems. We realize with new sequencing methods that there are viruses, when you just take some just nucleic acid samples from gut mucosa or respiratory mucosa and you just run it through a sequence, you find all kinds of sequence that clearly looks viral in nature. That is, you exclude the DNA of the host, or the nucleic acid of the host, and you amplify the rest, and you see all kinds of clearly viral sequences. But those same samples, when you try to grow them in tissue culture or even grow them in an experimental host, you don't get progeny out. So it seems like there's a lot of virus there, but we never saw it before because it's not growing on experimental hosts when you do the isolation, and also because it's not really causing disease. So we have to ask this question, are these new agents, if they're not pathogens, or maybe they are pathogens, but they're causing pathogenic effects in very subtle ways? And as we'll discuss, there's a number of diseases that now we suspect a partly infectious agent. So maybe just in some cases, in people with some genetic background, a virus contributes to disease. Or maybe they're neutral, or maybe they're even helpful. I mean, certainly many bacteria are helpful. Many gut bacteria, we're dependent on gut bacteria. Right? And as I said, this is true of bacteria in both cases. First of all, that there's lots and lots of them that fill a variety of roles in our ecosystem, in our personal ecosystem as well as the wider ecosystem. And there are many bacteria that cannot be grown in pure culture. We know that they're there, but there's no agar that they grow on readily. So what we can conclude now is viruses are everywhere. We're exposed to thousands and thousands of viruses every day in the air, in food, on fomites, inanimate objects that carry viruses. We can conclude first of all, from this, that we have a really good immune system. We must, because we're not sick all the time. Second, we also must conclude that while we may have a terrific immune system, probably most of these viruses aren't pathogenic anyway. Now, it may be that the reason they're not pathogenic is because we have a terrific immune system. Certainly we see when people have a defect in their immune system that things that don't make other people sick make them sick. And third, we can conclude that viruses are yet another extraordinary evolutionary success story. Many of the things that are fascinating about viruses aren't just the disease they cause, but how they've evolved and how hosts have evolved in return, because as with all organisms, none of us are in isolation. We're all part of somebody else's environment, and we're selective pressure and we're responding to their selective pressure. We need to address the question about whether or not viruses are alive. And the answer is a resounding and unequivocal no. Viruses have no metabolism. They have no homeostasis. the illustration I have here is of Tinker Toys, which is sort of an old-fashioned toy. I could have had Legos, I suppose. But the reason I have the illustration of Tinker Toys is that if you make an interesting structure with a Tinker Toy, you put it together. If you want to make a replica of that interesting structure with a Tinker Toy, you have to put another one together. And if you want to make 500 or 1,000 more, you got to put 500 or 1,000 more of them together. That is, you can't just leave your little interesting structure on its own and expect it to produce more of its own, the way a living cell does. Viruses are just like that. They're the proteins that they cause the cell to produce, the viral proteins, assemble. Now they assemble on their own, but the virus has to be in the cell so the cell can make the protein, and then they might assemble spontaneously, but they can't reproduce themselves through any kind of cell division or mitosis. So what does a virologist do? Well, they do many kinds of things. A clinical virologist is involved in research that will facilitate diagnosis and treatment of disease. So of course there's a lot of biotechnology overlap there just in developing the tools for rapid diagnosis and of course for developing treatments, vaccines, and antivirals. A lot of virologists are really molecular and cell biologists, and they're really just using viruses as tools to understand what the cell is really doing, because the virus goes in there and there are just so many more virus particles. Everything that's made by a virus is made generally way more abundantly than anything the cell makes, so it becomes easier to detect what's going on. Some virologists are really epidemiologists. They study transmission of disease, they study incidence of disease. They want to know, how does a disease get from one place to another? Ecosystem management is something you might not expect to see on this list, but there have been several instances where viruses have been used as agents to control some other population. Rabbit pox viruses have been used in Australia to try and limit the virus population. There have also been viruses used as anti-pesticide agents, that is, insect-specific viruses. And then gene therapy. Viruses are great tools to add genes into cells. And so viruses just become a tool to do what it is you want to do to help a patient, that is to cure a genetic disease or some other disease. In conclusion, viruses are unique infectious agents. They're not alive, but they are capable of evolving. So I often say that viruses are on the cusp of life. We just have to accept that they don't fall neatly into any category. The diversity of viruses is something we're really just beginning to appreciate. Initially, the only viruses we could know about were the ones that caused serious diseases, and there were plenty of those to occupy us, initially. But now that we have deep sequencing tools, we can appreciate that we are alive in a huge sea of viruses, as well as we're appreciating the diversity of bacteria and a lot of other things we couldn't see that are all around us. And that lastly, viruses are tools for many different disciplines, that we can use these to understand lots of aspects of life. In our next lecture, we'll talk about what viruses are made of.